wonderful KCET champions. I hope all my dear children are doing absolutely great. So we always tell you about ABHYAS KCET Test Series lots of time, and we definitely, when we say that, we mean it. That every KCET aspirant who is up there should be having this test series with them for sure. Why? Because you need to have an understanding of how exactly KCET examination works. Listen children, it is not as difficult as any other competitive examinations such as JEE or NEET and things like that. It is definitely easy. And sometimes when things get easy, that's also when we, tremble up there. Listen to me carefully. When I say this examination gives you a question which is easy to moderate, sometimes you might feel that, oh my God, these questions are like anyone can do it. And then you might spot some questions which makes you feel that I should have paid more attention to the littlest, tiniest details out there. Probably I might have not lost my marks. And that is exactly what you're going to feel over here also. There are questions which are super easy. There are questions which will make you think. Okay? And make sure you are there throughout and let me know what is your score after solving the entire set of questions. I have put 2 homework questions as well, which I want you guys to solve, and I'll show you how this is exactly look. So this is the first one, and this is the free one, of course. So over here, this is part test 1, where these chapters have been included: sexual reproduction in flowering plants, molecular base of inheritance, human production, and reproductive health, principles of inheritance and variation, and also evolution. When we say part test, what do you mean by part test? Part test means only few chapters are going to be merged up over here, but again, you are going to get 60 questions. Okay, and the questions and everything, it looks like this. I'll show it to you. So there will be a question, there will be an answer, and it will be given to you in test format. Okay, it is not going to be like this. PDF will be shared, but again, it is given to you in test format. Answers for this written elaborately as well. So this is what is generally up there. And now, let's go ahead. Let's see what's our score since we have just finished second PUC. So I'm pretty sure most of you would be able to do a lot of questions. So have it. Make sure you try this out. This is going to be super fun and super helpful for you. Okay, and always remember, only 1 minute per question. So here also we are just going to take 1 minute per question, not beyond that. Okay, so the center of each young microsporangium is occupied by microsporangium. That means basically talking about inside the anther. Remember, you had microsporangium where outside it is epidermis, endothecium, then you had middle layer. Inside that, the innermost thing, the centermost thing, what is there? So if at all you think like this, you have the epidermis. Okay, inside that the endothecium, inside that basically you have the middle layer. Inside that, what do you have? You basically have the next layer, which is known as tapetum, and finally you would be having something like this, which is known as sporogenous tissue. Center of each microsporangium. So this is one microsporangium. Similarly, you will have one here. Okay. Similarly, you will have one here. Similarly, you will have one here. Center of all of them, Baba, is known as what? It will be known as the sporogenous tissue, where what is going to be made? Your pollen grains are going to be made in future over here. So B is your answer for this particular question. I hope this is super clear to you. Yes, so it looks like this. This is your microsporangia. Center of all of them is basically sporogenous tissue. Fine. Filiform apparatus is present in. Filiform apparatus, Baba, where do you find filiform apparatus? In the female gametophyte. Remember, in the synergids. Synergids are there and the egg cell is there. Both of them, they are best friends, right? And now, the female gamete and the male gamete wants to fuse. Male gamete is coming through the pollen tube. Who is there to guide the male gamete inside? It is basically the filiform apparatus, right? And where do you find the filiform apparatus? In the synergids. That's why you call them. Synergids are the best friends of egg cell. Why? Because they have filiform apparatus which guides the pollen tube such that the male gamete and the female gamete can fuse. Okay. So please understand, it's a part of synergids. And you know what? I'll tell you one story. The synergids lose their life while the pollen tube goes to them. So it's like it's dying for its best friend out there. One synergid gets degenerated when basically a pollen tube is being formed out there. That's pretty sad, right? Yes. So that is the answer. B is your answer. I hope we are solving questions in that particular range, only 1 minute per question, right? And by the way, it's not just ABHYAS Test Series, and ABHYAS Test Series is available for rupees 999 now. It's a great offer that is up there. It was available for rupees 1499 before, but now it is available. Make sure you make the best use of it. And for children who feel that I need a crash course, not just a test series, there is KCET crash course, which is strictly online, and it is available for rupees 2499. Your PU1, PU2, entire thing, one-shot videos will be there, syllabus coverage will be there. Apart from that, you will be getting mock test. Apart from that, you will get PYQ in test format as well. Lakshya KCET offline crash course is also there, and there's a 3000 off going on. If at all you're interested in any of those, make sure you go through the links which are there in the description, only if at all you're interested. As of now, we are over here, we are dealing with ABHYAS. Yes, we are practicing. Okay, ABHYAS means practice. Now question number 3, monoecious plants with unisexual flowers. Monoecious plants means what, Baba? Monoecious means ones which have both the female and the male parts. Female and the male parts. Now a flower can be like this, where the female part is here and the male part is here. What do you call this flower as? You will call this as bisexual flower, isn't it? Yes, because both the sexes are there. Now there can be a flower like this which is having only the female part. This is another flower like this which is having only the male part. What do you call these type of flowers? You will call them as unisexual flowers. Now think about it. Suppose you have these type of flowers on a plant. That means you have both the sexes in one particular. So I'll say it's a bisexual plant and it has bisexual flowers also. Yes, yes. Okay, bisexual plants because it has both the sexes. In the flowers also, they are also bisexual. Why? Because it has both sexes. Now, one more scenario can be there where these type of flowers are one over here or one over here. What do you mean by that? The plant, Baba, is still bisexual. Why? Because it has the female part and the male part. But what about the flowers in there? They are unisexual. So it's a bisexual plant, but it has unisexual flowers. That is also known as monoecious. So the first condition can be like this, or it can be like this. Anyway, if the plant in general, I'm not talking about the flower, I'm talking about the plant. If plant is bisexual, basically you say that they are monoecious plants. They are what? Monoecious plants. Here you have been asked monoecious plants with unisexual flowers. That means that the condition is going to be like this. They have flowers like this and this, not together. Okay. Now, prevents autogamy but not gytenogamy. Can they prevent autogamy? Yes. Autogamy means what? From here to here when it goes. Arey Baba, it does not have the male part. So do you think that can happen over here? Self-pollination definitely cannot happen over here. So prevents autogamy but not gytenogamy. Gytenogamy means what? Basically, I have a flower over here and I have a flower over here. This is the male flower, this is the female flower. From here to here, on the same plant but between two flowers, if pollination is happening, that is known as what? Gytenogamy. So prevents autogamy, yes, it is preventing because it does not have a situation like this, but not gytenogamy, that itself is correct. So your answer is going to be A. What about the other options? Prevents allogamy but not autogamy, not at all true. Prevents gytenogamy but not xenogamy. Xenogamy means cross-pollination. Cross-pollination will definitely happen. Gytenogamy is also going to happen. Prevents autogamy and gytenogamy, that is also not possible. Your only answer for this question is going to be A. Okay? Yes. Perfect. So what's the score? Is it 3 out of 3 by now? Let's see. You have to tell me the actual score, okay, towards the end, and literally be honest there. Now, the mitotic division that occurs in generative cell results in the formation of generative cell. You know, the pollen grain which is there, it has vegetative cell which is going to be one big cell out there with irregular nucleus. Inside that you have generative cell with elongated nucleus. This generative cell, Baba, undergoes mitosis to produce what? That is the question. It is there to produce what? The two male gametes. Your answer is going to be A. But always read the other options as well. Vegetative and male gametes, no Baba. Vegetative cell and the other generative cell, no Baba. Generative cell, no Baba. Your only answer for this question is going to be A. Got it? Yes. Easy question, right? Now, the single large shield-shaped cotyledon of a monocot embryo is a monocot embryo. So, all this information is chumma for you to get confused. You just need to understand what is the name of the cotyledon for the monocot embryo. We all know monocot just has one cotyledon out there. Okay, rest everything is the embryonal axis. This part is the plumule, this part is the radicle. Above the plumule, you have the coleoptile. Below the root, you have the, below the radicle, you have root cap. And and as well as colorizza, and there is only one cotyledon out there, and you call this as what? Scutellum. What do you call this as? You will call this as scutellum. Why do they have one cotyledon? Because little amount of endosperm is still persisting. If at all endosperm was not there, it might have had two cotyledon, because for nourishment you need something, right? So either cotyledon or endosperms provide the nourishment. In the case of monocot, only one cotyledon is needed, rest is done by the endosperm itself. So coleoptile, colorizza, radical, no. Your answer is going to be scutellum. It is going to be scutellum. Easy question out there. Okay, so over here, now obviously if you do not remember the term, it becomes difficult for you. Yes, so always know that this is going to be super important. So this is a seed. This is how a monocot seed looks like. In the seed baba, you will be having plumule region. This is the embryonal axis. This is something that is going to give rise to the whole embryo later on. Okay, so you have the shoot apex and then the coleoptile. You have the roots and then the colorizza, and towards one side you will be having the reserve food material that is known as scutellum, and that is being mentioned over here. Rest, extra reserve material, that is known as endosperm. I told you, yeah? Yes. So I'm not going in depth, since as I said, it's just for you to have an understanding of how exactly this test series is and whether it is going to be useful for you or not. Get it? So that is the most important thing. And also when you try to solve this question, obviously you feel that, okay, where am I standing in the whole KCET prep, right? So yeah. Next question, it's a homework question for you guys, and it's from the chapter Molecular Basis of Inheritance. If at all you know the answer for this, comment down in the comment section and you can write 6th A, B, C, D, whatever is there, okay? And write please homework. The X-ray diffraction data for DNA was produced by X-ray diffraction data was produced by Watson and Crick, Morris Wilkin and Rosalind Franklin, Frederick Mischer or Atman. It's a very simple question. You can definitely know it. I'm pretty sure you know it, yeah? If you do not, refer your NCERT. Okay. What are the major roles performed by RNA molecule in addition to acting as a genetic material in some viruses? So what is the other role of RNA which is there? Apart from, it is also genetic material, especially in viruses RNA is a genetic material which is not stable. It changes its shape all of a sudden again and again, which is why we are not able to find a cure for any of the diseases which are virus borne. It's very difficult because viruses, they change the genetic material very fast, right? And that is why they have it. That is why they have RNA as a genetic material as well. Okay, so you have been asked, apart from acting as a genetic material, what are the other functions of the RNA? It functions as messenger, is it? Yes. For that you have mRNA. It functions as adapter, is it? Yes. It basically, there is tRNA, if at all you know. There is tRNA which goes and reads the whole mRNA. On one side it is reading the mRNA, on the other side it is basically carrying the amino acid. Transfer RNA is also what you call it. It also has structural and catalytic roles. So basically, rRNA, which forms the ribosomes, and the ribosomes, my dear children, what are they doing? The ribosomes are coming together, helping in reading of the mRNA, making in what? The proteins are there. How are the proteins made? Amino acids comes together. There is someone to make a bond between these amino acids. Basically, in all of that particular process, your rRNA and RNA derived things are also being used, right? So definitely it has a role like that. Your answer is going to be A. It functions as structural role only during protein synthesis, wrong. It performs adaptive molecule function only during polynucleotide chain. It plays a key role in regulation of protein synthesis. Baba, your answer for this question is going to be this, because it has everything that is true. Yeah? Yes. Next, the additional OH group of every nucleotide of RNA is bonded by additional OH, okay? Additional OH group of every nucleotide of RNA is bonded to. So over here, listen. The ribose sugar which is there in the case of RNA and the one which is there in the case of DNA is kind of different. This is the first carbon, second carbon, third carbon, fourth carbon, and there's a fifth carbon. Similarly, first carbon, second carbon, third carbon, fourth carbon, fifth carbon. If you talk about DNA, over here in the second position you just have a H. But in the case of an RNA, the additional OH group is there. which makes it highly unstable, or basically not unstable, okay? And it is on the second position. It is going to be there on the second position. And now over here, in the case of DNA, you do not have this oxygen, which is why you call this deoxy. There is no oxygen up there, which is why it is known as deoxy. I hope you have caught it. So where is the additional OH group found, Baba? It is on the second carbon position of the ribose sugar. Please understand, this is a ribose sugar. This is going to be deoxyribose sugar because it does not have an OH group. Okay? Or basically O group. Cool? Yes. Next. The additional material required for packing of chromatin at higher level is. So listen to me. What is happening? You have your DNA and you have to make sure that DNA is being compacted and put inside the nucleus. Now your nucleus is just this much and you have to put inside the DNA over here. For that, your DNA is pretty long. It's almost 6.6 into 10 to the power 9 base pairs long, which is pretty big, right? So how do we do that? We take the help of some histone proteins, right? We take the help of what? Histone proteins. So there are 8 histone proteins, 4 in the front and 4 in the back. You call this as histone octamer. Octamer as in 8 of them. 4 plus 4, 8. Onto this, your DNA is going to wrap. And there will be also one more thing. There is another histone protein which is known as H1 histone, which is acting like a linker. It acts like a linker. So whole of this thing is now known as what, Baba? It is known as nucleosome. Similarly, again you will be having histone proteins out there. Again this wrapping is going to happen continuously. That is how you get something like this. It looks something like this. What is this structure that is being formed over here? This is known as the chromatin. It is known as the chromatin. Chromatin also, which is being formed over here, it is known as the bead on a string like structure. Bead on a string. String is basically your DNA itself, yeah? And the beads which are there, they are nucleosomes. Bead on a string like structure is something that is basically nothing but the chromatin itself. The chromatin which later condense more and they will form what? Something like this, which is known as the chromosome. Yeah? Okay. So now listen to me. The additional material required for packing of chromatin at higher level. You have made the chromatin, but you need more packaging. Okay, for that, what do you need? Histone proteins are done. DNA, RNA, everything is done. Basically, you would need the help of non-histone proteins as well. There are some other proteins also which are very much required for the packing process. And those are known as the non-histone proteins. So your answer for this question becomes what? A. It becomes A. Cool? Yes. 10th question. Transcriptionally active gene is present in. Transcriptionally active gene. That means what? You know, from the DNA, you get the RNA, especially hnRNA, and that is known as transcription, right? It is known as transcription. So when I say transcriptionally active, as in the DNA which is available for the whole process of transcription to happen. Where do you find it? Heterochromatin or euchromatin? Ma'am, I do not understand what are these terms. Listen to me carefully. I made the chromatin structure right over here. There are 2 types of chromatins. One which is loosely packed, where there is too much amount of space over here. If at all there is space, enough space over here, you will call this as euchromatin. If at all they are very tightly attached to each other, you will call this as what? Heterochromatin. What do you call this? Heterochromatin. So there is euchromatin and there is something that is known as heterochromatin as well. Since you have space over here in euchromatin, RNA polymerase can come and attach over here and do the process of transcription, which is why we say euchromatin is what? Transcriptionally going to be active, whereas heterochromatin is going to be what? Transcriptionally inactive, because there is no space for the enzyme to come and attach. So your answer for this question is going to be B, because you are being asked about the active molecule. Got it? Yes. So over here, the answer for this question is this. Here you have the euchromatin, the loosely packed one, and here you have the heterochromatin. Always remember, this is transcriptionally active, and also it is going to be lightly stained. It does not take up a dark stain. It is going to have a light stain up there. Done? Done. Perfect. Next, X-albuminous seeds are seeds in which? X-albuminous seeds are the seeds in which endosperm is present, cotyledons are present, endosperm and cotyledons are present, endosperm is absent. What do you think? X-albuminous means non-albuminous. X-albuminous means non-albuminous, as in basically, Baba, they will not have endosperm left. Okay, in them the endosperm which is there, which is being formed at the end of double fertilization or basically later, this endosperm is completely utilized by the embryo to form what? Cotyledons. So they do not have the endosperm left behind. That is why you call them ex-albuminous or non-albuminous. Examples, you have pea, you have groundnut, okay, the pea, the groundnut, everything they come under this category. Most of the dicots, they come under, dicot as in the one having two cotyledons. Because they have made two cotyledons, they usually come under this category. Exceptions are there. There is castor, which is a dicot, but it does not come under this category. Castor comes in different category. So please remember that, okay? So these type of small informations are there. Ex-albuminous seeds are the seeds in which endosperm is present, cotyledons are absent, endosperm and cotyledons are present, endosperm is absent. What do you think? So cotyledon is present, that option is definitely not there, right? So what we go with? Endosperm is absent. Yeah, that is the one that is correct. Perfect. Okay? Yes. Next one. Apomixis is what? Apomixis, Baba, is the formation of seed without fertilization. So usually what happens, you have the ovary, you have the ovule. The ovary will be transforming into a fruit post-fertilization. Ovule which is there, it will be transforming into a seed post-fertilization. But over here, fertilization is not happening. That is, the male and the female gamete, they are not coming together by any chance. No fertilization is happening. Okay, that is one thing. That is one thing. Second thing, if fertilization is not there, okay, if fertilization is not there, okay, so that means that there is no need of meiosis as well. If fertilization is not there and you want to form an embryo has to be diploid in nature. So usually when fertilization is happening, your N comes together, and that's when you get the 2N zygote that will be transforming into an embryo later on. But over here I am saying fertilization is not happening. That means that already the condition over here should be diploid itself. The condition which is already over here should be diploid only. Then only basically this particular structure can give rise to an embryo which is diploid. Otherwise it cannot. Okay, otherwise it cannot happen. How will you get a diploid thing? You need someone to make it diploid, right? But it might be already diploid in nature. If you have to make sure that this is already diploid in nature, you would also have to make sure that there is no meiosis that is taking place. No meiosis happens up over here, which is why the egg cell is always going to be diploid. It is not going to be haploid, it is going to be diploid, and the egg cell is the only one which will be forming the embryo. It does not need a male gamete out there. Yes, so can I say apomixis is development of embryo from the endosperm? Wrong. Development of embryo from syngamy means fertilization happening. Wrong. Development of embryo without meiosis and without fertilization. Correct. No meiosis is happening, no fertilization is happening. The egg which is there, it is transforming into a new organism or an embryo altogether. So your answer for this question is going to be C. Okay, got it? Yes. Next. Which of the following statements about sporopollen is false? Sporopollen is a part of your, if you talk about the pollen grain, Baba, you have the plasma membrane, then you have the intine which is made up of Priyanka Chopra, that is pectin and cellulose. Then you have the exine which is made up of what, Baba? Sporopollen. It is the most toughest material out there. Okay, sporopollen. Fine. Exine is made up of sporopollen. This is correct. What do you need to find out? False statement. So always when you have to find the false or the correct one, underline what you're looking forward for, because sometimes we try to find something else, okay, between all of this thing. So exine is made up of sporopollen. Obviously, yes. Sporopollen is the most resistant organic material. Yes, it is organic. Look out for these terms. Sometimes they can give you inorganic also. It is organic. Okay, exine has apertures called germ pores. So these places that you see is known as germ pores, where the sporopollen is present. No, Baba, here sporopollen is not present, which is why you have pores in there, right? So over here, this is going to be a false statement. Sporopollen can withstand high temperature and strong acids, definitely, yes. So your only answer which is wrong is going to be C. Okay, perfect. Next question. Identify the wrong statements regarding the post-fertilization development. Post-fertilization, I told you, the ovary which is there, it will be developing into a fruit. The ovule which is there, it will be developing into a seed. Okay, the ovule has two things, that is the integuments. What happens to them? They become the seed coat. They become the seed coat, and they have specific name also. The outer one which is there, it is going to be known as testa. The inner one which is there, it will be known as tegmen. It will be known as tegmen. So testa is the outer one, tegmen is the inner one. That is there. The fruit, when I say ovary becomes the fruit, you need to understand that ovary wall which is there, you will now call this as a pericarp. Not the ovary. The walls of the ovary will be now known as what? Pericarp. Okay? Yes. The ovary wall develops into pericarp, correct. The outer integument of ovule develop into tegmen. The outer integument, Baba, this outer integument which is there, it forms a testa, not the tegmen. Testa is the outer one, tegmen is the inside one. Okay, yes. So this is going to be the wrong statement. You have to find the wrong statement, right? The fusion of nucleus develops into the endosperm. So when three nucleus, that is basically the two polar nuclei and one male gametes nucleus fuses together, it's triple fusion, and that leads to the formation of endosperm. That is correct. The ovule develops into seed. That is also correct. So you just have one option, and that is this. Tegmen is inside. Okay, the men are basically inside, and testa, which is there, it is outside. Okay. Next thing, fully developed female gametophyte in angiosperm is. It is a very simple question. Fully developed female gametophyte. You have 3 cells over here. You call them as what? Antipodal cells. Antipodal cells. You have 3 cells over here. 2 of them are synergids, and one of them which is there, it is known as the egg cell. Egg cell, and this is the synergids. Let's count now. Numbers. 1, 2, 3, 4, 5, 6. 6 cells now. 6 cells now. How many nucleus? 1, 2, 3, 4, 5, 6. 6 nucleus also. 6 cells, 6 nucleus. Done. Now what? There is a very big cell that is present over here. This very big space that you see is nothing but a very big cell. What do you call this cell as? The central cell. The central cell. So basically one more cell I'm adding up. Now how many cells do you have? You have 7 cells. Okay. The central cell, Baba, will be having 2 nuclei. Will be having 2 nuclei. What do you call them? You call them as the polar nuclei, right? The polar nuclei. So over here, if you talk about the nucleus, I have to add 2 of them. So 7 cell is there, but 8 nucleus will be there. One cell has 2 nucleus over here, then that is the central cell. So 7 cell and 8 nucleate condition. So that is, your answer is going to be D. Ayo, where is it? Yes. Got it? Perfect. Next question. In DNA, 10% guanine is present. How much thymine is being present? Okay. In DNA, so here they are not mentioning single-stranded, double-stranded. So we assume that it is going to be double-stranded, right? Double-stranded DNA, the rule is that, the Chargaff rule says that A is going to base pair with what? T. G is going to base pair with C. So how many number of C will be there? That many number of G will be there. How many number of A will be there? That many number of T will be there. And vice versa, right? Now over here, you have been given that guanine which is there, it is basically 10%. Okay. How much is the amount of what? How much is the amount of thymine is being asked? Okay. So let's find everyone. Thymine also, adenine also, everything. Listen to me carefully. If this is 10%, this guy is also going to be 10%, right? Both of them equal. Done. So 10 plus 10, if at all you calculate, it becomes what? 20% are them only. And what is the maximum percentage? We have 100. We have 100. So if at all you want to find the number of these two, what do? Basically, we are going to minus the 100 minus 20. 100 is there, 20 is being occupied. 100 is the maximum, 20 is being occupied by them. What is left behind? You have 80%. Now, do you write 80 over here? No, Baba. 80 is a whole. 80 is the whole thing that is left. So you have to divide them into 2. So here it will be 40. Here also it will be 40. What is your question? How much amount of thymine is present? So your answer for that particular question is going to be B, 40%. Got it? Yes, that's all. Next question. Which of the following is the correct with reference to the property of DNA? Okay, you have to find which one is correct. Okay, it should be able to replicate, definitely. It should evolve fast. If at all fast evolution was there, probably the earth would not have looked the same. Obviously, there is gradual changes, not fast. It is slow. It is not fast. So this particular statement is under scrutiny. Let's go ahead. It should be biochemically unstable. Obviously, this particular statement is going to be wrong. Okay, it should be able to mutate quickly. No, Baba, that's not true. You cannot allow it to mutate quickly. It's a genetic material. It has to be stable. It should not be undergoing faster mutation. It should be slow mutation. So the only correct statement that we have over here is what? It should be able to replicate. Simple. Yes. Next question. Match the column 1 with column 2 by choosing the correct combination of alphabets from the choices given below. Promoter site, terminator site, template strand, coding strand. Promoter site, if at all you talk about the transcription unit, you know there is a promoter site, there is a terminator site. Onto the promoter site, basically it helps in what? Initiation or starting of the transcription. So I'll say A goes with R. Terminator site, basically B goes with S. Can I find my answer with this? A goes with R and B goes with S. I just have one option that goes like that, and basically with that I can find the whole answer. Template strand shows the polarity 3:5. Yes, template strand is this strand, and basically the one that shows the polarity 3:5 dash, that is going to be template strand, which is going to undergo transcription. Whereas the opposite one, it is going to be known as coding strand. It is not going to undergo any transcription out there. Please remember that as well. Works? So yes. So template strand goes like this and coding strand goes like this. Perfect. Next thing. The functional unit of DNA molecule that codes for a particular gene product is the functional unit of a DNA molecule. So when I say it's a DNA, and I'm talking particularly about the functional unit. You know, DNA has multiple genes. In this gene also, there might be a particular portion which is going to actually code for some product, that is basically a protein. There might be some other sequences out there, promoter sequence, this sequence, that sequence. Okay, even though it's a part of gene, I'm talking about the functional one which actually forms the protein. It is known as cistron. It is known as cistron. And some of the organisms, like in eukaryotes, eukaryotes such as plants, animals, all of them, fungi, we have monocistronic nature. That means we will have one functional part which codes for one functional protein out there. In the case of prokaryotes, okay, that is bacteria, they will be having polycistronic nature. That means here itself they might have multiple cistrons coding for what? Multiple proteins out there. That is known as polycistronic nature. Here in us, it's not like that. You know, ZYA, black operon, Z gene, Y gene, A, all of them are together. That is polycistronic. P for prokaryote, P for polycistronic. But us, we are monocistronic. Anyway, the functional part is known as cistron. Next question. At 5' end of the polynucleotide chain, what is going to happen? At the 5' end of the polynucleotide chain, CHO group is attached, phosphate group is attached, pentose sugar is attached. H bond is present. What do you think is the answer for this particular question? At the 5' end, phosphate group is going to be attached, right? The 5' end over here, Baba, if at all you see, this is the 5' end. Here what do you have? You have the phosphate group being attached. And there will be free 5' end also. There also you will have phosphate group being attached. That is for sure. That is a sure thing that is going to happen out there. Yeah? So phosphate group is the one that is going to be attached at the 5' end. And if at all there is another sugar or basically another nucleotide at the top, basically the 3' end of that and the 5' end of this will be attaching with the help of the common phosphate group. That is known as phosphodiester bond. Okay. Now, select the mismatched pair. Okay, you have to find the mismatched pair. Microsporangium is basically known as the pollen sac. Is it? Yes, because that is where the pollen grains are going to be formed. Megasporangium is basically known as the ovule. Correct. It is actually known as the ovule. Pollen grain is known as the male gamete. No. Pollen grain is known as the male gametophyte. It is going to be known as the male gametophyte. It is not the male gamete. Right? It is going to form the male gamete. Embryo sac is the female gametophyte. This is correct. So your answer for this question is going to be what? C. Cool. Understanding, everyone? So as I said, I am going fast. Why? Because this is supposed to be a demo lecture. Okay. So entirely when in Abhyas, when you go, you are not going to find the video solutions with it. You have to solve the question by yourself. You will be having the PDF where the answers and everything will be written. It's sorted. It's all clear. Okay. So have that thing. You have to solve the questions from your end itself. It's a mock test. And mock tests have to be given alone by you guys. Yes. So I'm just here to guide you, to help you in some ways possible. That's all. So the given diagram shows microsporangia of a mature anther. Identify A, B and C. So what is A, Baba? Over here, look at this picture very carefully. The A goes basically inside. It's not the outer layer. This is the outer layer. Outer layer is known as epidermis. Inside that, what do you have? You have the endothelium. Inside that, what do you have, Baba? You would be having the middle layer. Inside that, what do you have? You have the tapetum. Okay, so endothelium, middle layer and tapetum could be the answer for this particular question. So A is endothecium, B is middle layer, and C is tapetum, which is why your answer for this particular question is going to be C. Okay? Yes. Now, select the correct option regarding the ploidy level of different structures of an angiospermic ovule. Nucellus, MMC, and functional megaspore. What is the ploidy of it? Listen, if this is the ovule, there are normal cells that are surrounding over here. What do you call these cells as? These cells are part of nucellus. And basically, the cells are normal cells. So I'll say it's diploid in nature, right? And then over here, you have one big cell which is known as your megaspore mother cell. This is what megaspore mother cell. This is also going to be diploid. It is also going to be diploid. This diploid megaspore mother cell which is there, it undergoes meiosis. The first strand of meiosis, meiosis 1, becomes haploid in nature, again undergoes meiosis. Again, it will be haploid in nature towards the end. This will be what? Meiotic 2, meiosis 2, right? Now, at the end of it, you're getting haploid cells. You're going to get haploid cells. Now, 4 of them are not going to be active. 3 of them are going to degenerate. Only 1 remains functional, and that is known as the functional megaspore, which later forms the female gametophyte, that is the embryo sac. Now, over here, you have been asked about the ploidy. The nucellus, you saw it is diploid. You just have 2 options like that. Cancel the other options. Okay, MMC, again, it is also going to be diploid. Just have 1 option like that, and the rest is history. So, functional megaspore is definitely going to be haploid in nature. Right? Right. Perfect. Next, given below the events that are observed in an artificial hybridization program, arrange them in the correct sequential order and select the correct option. Artificial hybridization basically means that I have a plant and I have another plant. I know both of these plants have some unique good features. I need to make sure that both of them breed together to produce the plant that I want, the hybrid variety. That might be good in all sense. Okay, so what do I do? First of all, I have to select the parents, right? First process would be to select the parents. First of all, let's read out all the options. Rebagging, bagging, emasculation, selection of parents. What is it? I think, okay, it is dusting. Dusting the pollen on the stigma, collection of the pollen from the male parent. Okay, so let's start. First thing that you do is selection, right? Selection is 2. Option number 2, so I'll cut off this one because this here it's written 5. When you cut off that, before that read it. It's emasculation, obviously that is not going to be the one. Okay, so I have 3 options now. After selection of parents, what would I do? After selection of parents, if at all I see that basically this particular parent which is there, it has both the male and the female part. Okay, and I just want the female part. I will cut off the male parts out there because I do not want their pollen grains to land up over here. What do you call this process as? Emasculation. I will call this as emasculation. I want to protect the female plant out there, right? So after 2 I have 5th. After 2 I have 5th, I have got this particular option. Let me see. I'll go according to this option now and let me see if at all the answer would be correct. Okay. After emasculation, basically 3. 3 is what? Bagging. Okay, so once I cut it, I have to make sure no other pollen grains will be landing over here, which is why, what do I need to do? I need to bag this whole setup up. So I will cover this up. This is known as bagging. This is also correct. Fine. After that, what I have to do? 6. Collection of pollen from the male plant. From some other male plant, I have to collect the pollen grain because I have cut it anyways. So I have to collect from the other place. That is also correct. After collecting, what do I have to do? I have to basically dust it. Okay, dust a pollen on the stigma, correct? That is also correct. After dusting, what do? I cover it back. I am going to cover it back. Why? Because I do not want any of the pollen grains to land on it. So, which is why your answer for this question is going to be B. So, first try to spot off your answers and then with the help of options itself, try to find the answers. You get it? That is the logic out there. So, your answer for this question is going to be B. Cool? Yes. Next question. How many meiotic divisions are required for the formation of 100 functional megaspores? How many meiotic divisions are required for the formation of 100 functional megaspores? So, you know, 1 MMC I told you, it's 2N in nature. It undergoes meiosis. You get 2 cells, haploid, haploid. It again undergoes meiosis. You get 4 cells at the end, N, 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 N. But 3 of them get degenerated. Only 1 is there. So, if at all I am saying I want 100 of them, 100 of them should undergo meiosis. If I want 100, because only 1 produces one. Okay, one megaspore mother cell is going to give me only one functional megaspore, right? So basic 100 of them will be giving me 100 of them. You get the logic? So how many meiotic divisions are required? So when I say the number of, when I get to understand the number of, the megaspore mother cell, basically I get to understand the number of meiotic division also, because they undergo one meiosis to do all this thing, right? So if 100 of them are there, they are going to undergo 100 meiotic divisions out there. So that's the basic logic. So your answer for this question is going to be A. I'll explain once more. One megaspore mother cell gives rise to one functional megaspore with the help of one meiotic division. Meiosis happens in two phases. That does not mean it's two meiosis. Only one meiosis is there, right? Yes. Now when I'm saying I have to produce 100 of them, that means 100 of them are going to undergo meiosis. How many times? Only once. One meiotic division. Okay, you get it? That's all. So your answer for this question is going to be A. Perfect. The answer is written here as well, if at all you just want to take a look at it. Okay. Next, splicing is meant for eliminating what? Splicing means what? You have your DNA has some regions that do not code for anything, some regions that code for something. You call this as introns, and you call the coding ones as exons and the non-coding ones as introns. They are intervening sequences. That is why they are known as introns. So when I do splicing, why do I do splicing? This DNA is going to transcribe into hnRNA. It will also have the same thing. The ones that do not code, the ones that code, the ones that do not code, the ones that code. But when you say that you have to form an mRNA, the mature RNA, which will be forming the proteins or coding for the proteins later on, you have to make sure only the coding part is there. So what do we need to do? We have to remove this part, and the process that it undergoes is known as splicing. Removal of introns, they are known as splicing. Splicing is meant for eliminating introns. Your answer for this question becomes D. Cool? Yes, ma'am. Next. Assertion. hnRNA is larger than mRNA. Baba, you see, hnRNA has introns, exons and everything. So obviously it is going to be larger than mRNA, because mRNA you cut it off and you have specific sequences, right? So I'll say hnRNA definitely is going to be larger than mRNA. Correct. Reason. hnRNA has, first of all, so when I get to understand that the reason is correct, I look over here. Where is it? Okay. Both assertion is true, assertion and reason are false. So basically this option goes off because I know assertion is definitely true. Okay. Now, hnRNA has non-translating introns and more exons than required for translation. Okay, until here it was correct. hnRNA has non-translating introns. But does it have more exons than required? No, definitely. It has the required number of exons only. Right? So that particular statement would be wrong. So can I say assertion is true, but the reason is false? Yes, I can say that. So if you do not read your options carefully, you're getting your answers literally very, very wrong. So make sure you do it carefully. Next thing, find out the wrong statement among the salient features of the genetic code given below. The codon is triplet. Is the codon triplet? Yes. What do you have to find out? You have to find the wrong statement. 64 codons code for 20 amino acid. Is it? And 3 codons do not code for anything. Wrong. Wrong. 61 codons which are there, they code for the amino acids, 20 amino acids. 20 amino acids. And the 3 codons which are there, they do not code for anything. They are known as stop codons. Together it forms what? 64 codons are there. So can I say the codon is triplet, 64 codons code for 20 amino acid, and 3 codons do not code for any amino acid? Wrong. This particular statement itself is wrong. But here you have multiple options, so you have to be careful, right? Next thing, genetic code is nearly universal. Yes. So whatever the code is coding over here, that itself is going to be coded in bacteria and everything out there. So this particular statement is correct. The code is degenerate because some amino acids are coded by more than one codon. Is it? The code is degenerate. Degenerate as in, for example, UUU, UUA. Every one MABA is going to code for phenylalanine only. Multiple codons are there coding for just one amino acid. That is what you mean by degenerate. The code is degenerate because some amino acids are coded by, some amino acid, that is phenylalanine, coded by more than one codon. Correct? That statement is correct. The codon is read in mRNA in a contiguous fashion. Is it read in contiguous fashion? Yes. So there is no full stop, comma and everything. Everything is being read in a contiguous fashion. 3, 3, 3, 3. So only statement which is wrong would be B. One only. Got it? Yes. Next. Identify A, B, C and D in the below given diagram. So this is the picture that is given to you. You have to find what is A. A is where? Yeah, okay. A is over here, B is here, C is here and D is here. That means what is Z coding for, that is what is Y coding for, what is A coding for, so D coding for. So you know the one that attaches with the repressor protein is going to be known as what? A inducer. You call this as inducer. Now, there are different types in inducers. What are the different types of inducers that you have? You can either have lactose as an inducer or allolactose as an inducer, right? So A is definitely going to be either inducer or lactose. So in this option, repressor is given. In this option also something repressor is given. So I just have these two options. With A is inducer, A is lactose. Okay. B is what? B is going to be beta-galactosidase enzyme, right? C is going to be permease enzyme. D is going to be transacetylase enzyme. B is beta-galactosidase. This is wrong. So that is wrong. So your only answer for this particular question becomes what? This. Got it? Your only answer for this particular question is going to be C. So if you do not know the functions of this, basically you will lose mark up over here. So as of now, when I'm solving with you, you feel that it's easy. But you need to know the basics, otherwise you might mess around. Deciphering of genetic code was done by, any idea? So basically, who was behind the understanding of genetic code and everything? Nirenberg and Mathai, Hawley and Kornberg, and F.H.C. Crick, and both A and B. What do you think is the answer for this? Who brought in the knowledge for genetic code? It was Nirenberg. Okay, it was Marshall Nirenberg and Mathai. So your answer for this question is going to be A. Very, very important piece of information. Again, these scientists are not being given too much amount of. Hargovind Khurana is also up there, by the way. So anyways, so this is a very important piece of information which we usually miss. We remember Francis Crick, Maurice Wilkins, this, them. A lot of things, but we forget the names of the people who are behind genetic code. Make sure you know that. Yes. By the way, we have solved 30 questions, 30 more left. Let's go. The given figure shows male reproductive system. Some structures are marked as A, B and C. So A is given over here. What is A? You can clearly see A is semiannular cycle. It's a gland that is going to help in production of fructose. Okay. And B is basically what? B is going to be uterus. It feels like it is uterus, yes. And C basically it is bulbourethral gland is what I'm understanding because it's not too much clear. Yes, and D is going to be the testis. Okay. Identify the structure whose removal will affect the lubrication of penis. That is the question. Okay, so which is that structure? If at all I remove it, that is going to mess around with the lubrication of penis. For that, we need to know the function of each of them, right? Over here, A is basically semiannular cycle. I told you it releases fructose. Fructose is providing energy. B is basically, I don't know what have they mentioned. Okay, so I think the line goes like this. So it is basically the prostate gland. Prostate gland is based for production of it is there for production of calcium as well as the enzymes out there. Okay, calcium as well as the enzymes out there. So both of them are required for fertilizing capacity and things like that. It has nothing to do with the lubrication of penis. Now over here you have C option, which is basically bulbourethral gland or Cowper's gland, and that is there for lubrication of penis, which is why your answer for this question is going to be C. Now do not look out for C option over here. Here by chance they have given C as C, but they can give you ulta also. Here they can put B and here it's C. So look out for this particular thing. Sometimes we mess around that also. Be careful up there. It is going to be C. Okay. Yes. Next, given below are two statements. One is labeled as assertion and other is labeled as reason. Okay, assertion says amniocentesis for sex determination is one of the strategies of reproductive and child health care program. Okay, reason, ban on amniocentesis checks increasing menace of female feticide. So was the ban put on amniocentesis because the female feticide was increasing? That is, female killing was been increasing? Definitely yes. Is amniocentesis for sex determination one of the strategies of reproductive and child health care? It is not for sex determination. It is amniocentesis was basically introduced for finding the genetic abnormalities out there. So in that particular case, what will be your answer? Assertion is false, but the reason is true. Amniocentesis can be used for sex determination, but it is not for sex determination in reproductive and child healthcare program. It was for what? It is for finding the genetic abnormalities. That was the reason why it was being introduced. But then there's a ban being put because of the increasing female feticide rates which was being observed. So D is your answer for this particular question. Cool? Yes. How many different genotypes and phenotypes are produced in F2 generation of a dihybrid cross performed by Mendel's, considering that the parents are true breeding for round and yellow and wrinkled and green? So basically talking about the dihybrid cross experiment, and you have been asked what are the number of phenotypes and what are the number of genotypes that is observed. This is a very easy question. So let's go. We know the the ratio, the genotypic ratio, the phenotypic ratio is like this. Phenotypic ratio means physical appearance ratio. 9 of them are going to show the dominant parent character, that is round and yellow. 1 of them are going to show the recessive parental character, that is what? Wrinkled and green. Okay, now over here you have 3 and 3. What are they? They are going to show over here round was with yellow. I'll make round with green. This is a new character that is being seen. This is a non-parental character that is being seen. Now here I have wrinkled with green. I'll make wrinkled with yellow. This is also a new parental character. Anyways, 4 phenotypes are there. Yes, 4 phenotypes are there for sure. So I'll say this is a wrong option because 16 phenotypes are there. Now whenever you cross, you have seen that 16 progenies are being formed. 9 plus 3 plus 3 plus 1 is equals to 16. And you find, do you think that 16 genotypes of the genetic makeup is also there? No. There are repetitions of genotypes, okay? There is repetition of genotypes. That is, I mean that it can be 4 in number. The same genotype was there 4 in number and out of 16 or probably more. You get it? So what do you understand from this is that you have to just count the unique genotypes, not the entire genotypes. The unique genotypes are only 9 of them. And I remember it by this number, 9, the first letter 9. Get it? So your answer for this question is going to be this. 4 phenotypes and 9 genotypes. Cool? Perfect. Let's go. The theory of spontaneous generation stated that life arose from the living forms only. Spontaneous generation, okay? Okay. Life can arise from both living and non-living. Life can arise from the non-living things only. Life for us is spontaneously neither from the living nor from the non-living. Spontaneous generation said that if at all you have a sack of rice being kept there, and people actually saw that from the sack of rice, mouse were coming out. So they were like, from this non-living thing, a mouse is coming out. That is spontaneous generation. They thought like from that thing, this thing evolved or that has come. So this is basically, it means that life can arise from non-living things only. That was the logic behind it. But obviously it was being disapproved by the help of the Louis Pasteur experiment. Cool. Next thing. The given figure shows the diagrammatic sectional view of the female reproductive system with few structures marked as A, B, C and D. Over here A, B, C and D. So what do you do? First thing that you have to do is mark what is D. D is fimbriae. Mark what is C. C is what? Infundibulum. B is what? B is ampulla. And A is isthmus. I know this. Now you look out for that in the option. Is A mentioned as isthmus? No. Is B mentioned as ampulla? Yes. So have to take in consideration. C is mentioned as infundibulum? No. Isthmus is being mentioned over here. D is mentioned as fimbriae? Yes, this is also correct. Now you have to look for the things that is there. So among these options you have to look. Anyway, this and this has got cancelled, right? B is ampulla and it is a wider part of oviduct where fertilization occurs. Is that true? It is a wider part? Yes. And so infundibulum is a funnel shaped, B is the wider part and isthmus is the narrow part. And basically ampulla is actually the place where fertilization occurs. So that particular statement is also correct. Now fimbriae, it collects the ovum before ovulation. No Baba, ovulation has to happen, then only ovum will be there, no, to collect it, to being collected. So basically this is also is wrong. Only option that is correct is B. Got it? In this manner you save time. So label them first, then only go ahead. Okay. Next in, find out the correct matching. Correct matching is what you have to find out. Non-medicated IUDs lipase loop. First statement itself is correct. Only one non-medicated IUD is there, intrauterine device, which does not release anything inside the uterus, and that is lipase loop. Copper releasing IUD, you have CUT, CU7, LNG20. LNG20 is hormone releasing IUD, not what you call copper releasing IUD. Okay, the replacement for that is multi-load 375. Here multi-load 375 should have come. Basically that is copper releasing IUD. Okay, progestasert and LNG20 is hormone releasing IUD. So this particular statement is also wrong. All of the above definitely cannot be the answer. So the only answer for this question is going to be A. And this is how lipase loop looks. It is non-medicated. How does it function? Basically when you're putting something different from outside, it is foreign for your body. So your body is going to be immunologically very active, right? Now when the sperm, suppose insemination happened and sperm came inside, your immune system will be targeting those cells and will be making sure that they are phagocytized. They're taken inside the cell and are being killed. Why? Because your immune system is super active because of this particular guy. Okay? Yes. Now, read the following statements and identify the correct option. A. Pleiotropic genes have multiple phenotypic effects. Pleiotropic gene. Pleiotropy means basically one gene will be having multiple effects. You might have seen phenylalanine hydroxylase. This is an enzyme which is not present in the case of people who are having phenylketonuria. In them, since the enzyme is not present, phenylalanine is not getting converted, and which is why it is going to be accumulated in their body. That is one problem that is going to happen. Second problem that happens is that, this enzyme is responsible for the conversion of phenylalanine to tyrosine. And tyrosine is super important for our pigmentation, skin pigmentation, hair pigmentation and things like that. Okay, it is going to help in the production of melatonin and things like that. So basically, that function is also not happening, which is why you will be having reduced pigmentation. So two things are being hampered, two characters are being hampered because of one gene being affected. These genes are known as pleiotropic genes, genes which are affect on multiple phenotypic things. So this is correct. Multiple alleles exhibit same phenotypic expression. No, multiple alleles will be having multiple effects. IA is going to code for basically having A blood group, IB for B, and IO basically will not be coding for any sugars out there. So all of them have multiple phenotypic expression, not the same one. So this particular statement is going to be wrong. What do you have to find out? You have to find the correct option. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. What do you mean by that? For example, I have already told you the skin color, okay, the skin color and your height. All of them are going to be coded by multiple genes, not one gene. So usually in mental we have studied capital T gene is going to code for tallest. No, here multiple genes are going to show their effects for basically one particular character. Here the genotype will be capital A, capital B, capital B, capital C, capital C. All of them will be showing an effect together. So that is known as polygenic. Multiple genes are therefore responsible for one phenotypic character. So in a polygenic trait, the phenotype reflects the contribution of each allele. That is, effect of each allele is going to be additive. It is going to be added up. I hope that is super clear. Yes. So A and C are definitely going to be true. A and C are definitely going to be true and B is going to be incorrect. So your answer for this question is going to be D. Cool? Perfect. Next. Choose the pairs of organs that are a result of convergent evolution. Convergent evolution means they do not have a common ancestor. They are staying in the same habitat, which is why they have the same sort of functions. Structurally, they are not going to be similar. Functionally, yes. So functions are definitely going to be similar. Wings of butterflies and wings of birds. Function same for flying only. Convergent. Flippers of penguins and flippers of dolphins. Function is same. This is a bird, that is a mammal, but flippers are there for swimming itself. Correct. Eyes of octopus, eyes of mammals. This is invertebrate, this is vertebrate, but eyes are there for looking itself. So this is correct. Thorns of bougainvillea and tendrils of cucurbita. No, both of them do not have a similar function. Thorn is for protection. Tendril is basically for what? Tendril is for support out there. So they are not an example of conversion. They do not have a common function. Wrong. That is divergent evolution example. Sweet potato and potato. Both of them have the same function of doing what? Storage. They have evolved separately, but they have the same function of storage, which is why again convergent evolution. Vertebrate hearts and vertebrate brains. Vertebrate heart and vertebrate brains. What do you think is the answer for this? Convergent or divergent? Convergent or divergent? You tell me. So 1, 2, 3 and 5 is very easy for you to spot. 1, 2, 3 and 5 is definitely going to be easy for you to spot. But this you might mess up, right? What do you think? It is going to be divergent, not convergent. So basically your answer for this question is only this. Only this. Okay? Yes. Perfect. The first meiotic division during oogenesis is completed at this stage of. First meiotic division, Baba, where does it happen? You know, from the primary follicle, inside the primary follicle, you have the primary oocyte. Inside the secondary follicle also, you have the primary oocyte. Finally, inside the tertiary follicle is when the oocyte, the secondary, sorry, here also your primary oocyte, this is primary follicle, this is secondary follicle, and now this is tertiary follicle. Inside this only, the primary oocyte is finally going to divide into secondary oocyte and the first polar body. So it's in the tertiary follicle where this happens. So the first meiotic division during oogenesis is completed at this stage of primary oocyte within the primary follicle, wrong. Primary oocyte within secondary follicle, wrong. Primary oocyte within the tertiary follicle, yes. Okay, primary oocyte within the tertiary follicle, yes. Secondary oocyte within the tertiary follicle, no. So the answer for this question is only going to be C. Cool? Yes. Next. Saheli is a contraceptive drug. Select the incorrect statement about it. Saheli, is it a contraceptive drug? Definitely. It is. It is one of the best one. Oral drug, yes. What do you have to find? The incorrect statement. Non-steroidal, yes. It does not release any hormones. Okay. Once a month pill, okay. Is that correct? Wrong. Once a week pill, not a month pill. This is how you get your answers wrong. High contraceptive value, yes. So sometimes what happens is that we do not know whether it's non-steroidal or oral drug and things like that. But once a month, once basically that term we have seen, so we remember that. But you have to make sure you focus on the next thing also. It's once a week pill. Got it? Yes. Perfect. Read the following statements and choose the option with the wrong statement. In phenylketonuria, the affected individual lacks an enzyme that converts the amino acid phenylalanine to tyrosine. That is correct. You have to find what? The wrong statement. Possibility of a male becoming hemophilic is extremely rare. No. Males can have hemophilia. Males can have hemophilia. They need to have the X chromosome from a female. Now this female can be a carrier. From here also they can get one X chromosome is needed, and that can be got from a female carrier. For a female to have the disease, it's difficult. Why? Because a female, if at all you want a female to have the disease, one X chromosome basically should be coming from the father, another chromosome should be coming from the mother, right? Then only you will get something like this. Now, for a father to stay alive, now female, she is a carrier, she can stay alive because she has a normal gene. For father having one gene being affected, do you think he can be alive until the time for reproduction? No. Which is why there is no possibility of a girl baby also. You get it? There is no possibility of a girl baby having the disease because he might not be alive until that particular point of time. Got it? So for a female to be hemophilic, that is extremely rare. It's not the thing that is there for male. So this particular statement is going to be wrong. Sickle cell anemia is caused by substitution of glutamic acid by valine at the 5th position of the beta globin gene. Wrong. Yes, glutamic acid is being substituted by valine. G comes before V, so it was G, then it was replaced by V, but not at the 5th position. It happens at the 6th position. So this particular statement again is going to be wrong. Now, myotonic dystrophy is an autosomal dominant condition. Yes, it is the only autosomal dominant condition which is given in your NCERT in the form of the pedigree chart. Okay, so this is also correct. So 2 and 3 are the ones which are wrong. 2 and 3 alone are wrong. 2 and 3 alone are wrong. Your answer for this question is going to be B. Done. Perfect. Next, frequency of an autosomal lethal gene is 0.4. Okay, the frequency of carrier in a population of 2000 individual is. How do you find the answer for this? So listen to me. Frequency of autosomal lethal gene. So I am taking it to be Q. Q is equals to 0.4. Hardy-Weinberg law, you have to use that over here. We have to find what is P first. Okay, because we have to look for what? We have to look for carriers. Carriers means they will have one gene to be normal. So it's a heterozygous condition which is going to be there. One gene is going to be normal and the other gene which is there, it probably can have the effect. Suppose this is the one that is affected gene. Suppose this is P and this is Q. So you have to find the people who are heterozygous. And whenever you have to find heterozygous condition, you do 2PQ. That is 2 into A and A. you need to understand there is a formula. P square is plus 2PQ plus Q square is equals to 1. This is a formula. P square stands for homozygous condition. Capital A, capital A. Q square stands for small a condition, homozygous recessive condition. 2PQ stands for this particular condition. So you have to make sure 2 is being multiplied. So how do you find this? Listen to me very carefully. This is basically, if I make this condensed formula, it comes out to be P plus Q whole square is equals to 1. If I put the square root on both the side, it will be coming out to be P plus Q is equals to 1. If your Q is 0.4 and P plus is 1, then P will be what? The shift that side, 1 minus 0.4, P is equals to 0.6. Agreed? Agreed. Now you just have to replace it over here. 2 into 0.6 into what? 2 into 0.6 into what? Into 0.4? Into 0.4. What does it come out to be? It comes out to be, I think, 0.48. Yes, it should be coming out to be 0.48. Now, 0.48. How many individuals are there? 200. Out of 200 individuals, I say basically 48%. 0.48 is the frequency. I can also say 48%. Now, if I want to find out in 200 individuals, how do I do? Basically, I'll do multiplication. 0.48 into 200, that comes out to be 96. In population of 200, this much. Got it? Yes. So, it's nothing but this only. It's nothing. 48% of 200 is basically your answer, that is 96. Cool? Yes. Now, which of the following statements are incorrect? A rapid decline, I think that is one of the best questions out there. A rapid decline in the death rate is the only reason for the population explosion. What is incorrect? A rapid decline in the death rate. If people are dying less, that is the only reason for the population explosion? No, that's wrong. The marriageable age decided by the government is 18 years for the females and 21 for the males. Correct. An ideal contraceptive is the one that reduces the sexual drive of the user? No, it should not mess with that. The one-child norm has mostly been adopted by people living in the rural areas. One-child norm in the rural areas? No, Baba. For them to be educated is very difficult, okay, because of the location they are in. In urban areas, yes, people adopted the one-child norm because they got an understanding of it. In rural areas, that is not the condition. So this here, you miss it out, you get it? This is also wrong. So 1, 3 and 4 is wrong. 1, 3 and 4 is wrong. That means your B option is the correct one. Perfect. Next question. A normal woman whose father had hemophilia. If father had hemophilia, the condition was like this. That means a woman will also be having at least one X chromosome which is affected, and it is a normal woman. So that means she is just a carrier. She is a carrier. She is not having hemophilia. She is just a carrier. Marries a normal man. Marries a normal man. So let's make it over here. Now you have got the idea. Okay. Normal man means normal man only. No problem in that. And over here you have a female. Now what is the percentage? What is chance of hemophilia in the children? That is the question. Okay. X and X goes, you get normal X and X, no problem. This and this, X and XH, she is just a carrier. This and this, X and Y, normal male. This and this, one guy who is affected. One guy who is going to be affected over here. So out of 4, 1 is affected. That means 25%. 25% of the children are going to be hemophilic. They are going to be hemophilic. A is your answer for this particular question. Perfect. Perfect. Next one. Who disapproved the theory of embryological evidence as to the evidence of evolution? Who disapproved that embryological evidence is not true? Embryological evidence means basically, for example, when human beings are formed, in our embryonic stage, we have the gill slits, we have the tail and everything. It shows that we have evolved from what? The fishes. So it goes like this. Farm. Fishes, amphibians, reptiles and mammals. In between that, reptiles also gave rise to the birds. Always remember that. So in that case, we should have also shown the features of reptiles and amphibians also, is what Carl Ernst von Baer said. He said that that is not happening, which is why I will not approve this. Ernst Haeckel was the person who gave the theory of embryological evidence, and Carl Ernst, both of them are Ernst only, but the one with bigger name said is a big no. Okay? Yes. Next, hormones secreted by placenta to maintain the pregnancy are 4 hormones are there. HPL, human placental lactogen, HCG, human chorionic gonadotrophin, progesterone and estrogens. Progesterone and estrogen were basically stimulated by the ovary first, but now by them. So your answer for this question is A. Here, relaxin is given. Relaxin is released by the ovary during the later phase of the pregnancy for relaxing the pelvic ligaments such that the baby can go. Prolactin is being released by our pituitary, not by this. Okay, and yeah, glucocorticoids basically are released by adrenal glands. So, 47th, nearly dash MTPs are performed in a year all over the world. Any idea how many MTPs are performed? 45 to 50 billion, 45 to 50 million, 45 to 50,000, 45 to 50 lakhs. The answer for that is 45 to 50 million. A straight question from your NCERT. Sometimes we forget about these basic things. It is 45 to 50 million. Million, not billion, million. Now this is where Abhyas is of use. I'm telling you, it will be definitely useful for you. Study the pedigree chart given below, and it is available for just ₹999. You should be taking that up. What do you think? Is it just a promotion? What do you think? Is it just a promotion out there? Don't you think it will help you guys? Obviously, yes, right? So yes, take it up. Study the pedigree chart given below to identify the disorders. Sickle cell anemia, hemophilia, myotonic dystrophy, color blindness. What is this? So over here, first of all, myotonic dystrophy cannot be the one because it's a dominant disorder. And if it was a dominant disorder, you would be seeing at least one parent being affected in every generation. Here there's no parent being affected. Here also there's no parent being affected. But their kids are. Here also their kids are. So this is not the one. Now, you have to find whether it's sex-linked or autosomal. Hemophilia and color blindness are sex-linked recessive, and sickle cell anemia is autosomal recessive. In sex-linked recessive, you could see that in one generation itself, more males are the ones which are being affected. Here you see one female and male is being affected, like equal thing. Here also, okay, one male is affected, only one guy is there, okay. You cannot say that males are the one that are totally more affected over here, which is why I'll say it is an autosomal disorder. Autosomal recessive because it is skipping the generation for sure. And yes, so that is something that is up there. Please remember it. Okay? Yes. I'll be coming with more, rules for the pedigree chart. It will be more easy for you to identify then, but yes. Identify the correct sequence in which the various stages of parturition takes place. Okay, how does the parturition take? Parturition means expulsion of the baby. Shedding of the placenta, dilation of cervix, delivery of the baby. Dilation of the cervix, shedding of the placenta, delivery of the baby. Dilation of the cervix, delivery of the baby, shedding of the placenta. You might have seen that first the cervix gets dilated, that's when the baby has to move out. After the baby, the placenta comes out. After the baby, Baba, you have been pulled, the placenta has been pulled out. You get it? So your answer for this question is going to be C. Dilation of the cervix so that baby can come out, delivery of the baby, and then the shedding of the placenta. Cool? This is a logical question, not there much in your NCERT, but yes, you should know it. Now, how many technologies in the list given below are showing in vitro fertilization? In vitro means in the laboratory condition. That means outside the body. Write it. In vitro means outside the body, in the lab. Make it clear. Now, gift. Gift means you are transferring the gamete inside the body. Fertilization is going to happen inside only. Wrong. Zift. Over here, you have the zygote being formed outside. That is why zygote intrafallopian. You're putting the zygote. So obviously it is put in the, it's done in the lab. So second option is correct. ICSI. ICSI means intracytoplasmic sperm injection. Obviously, if at all you want to insert the sperm inside the cytoplasm of the egg, you have to do in the lab only. That is also IUT, intrauterine transfer. What are you transferring? You are not transferring gametes, you're transferring embryo, which is more than 8 blastomia stage. Again, you put it. AI, artificial insemination. Artificial insemination means you are giving the sperm and putting inside the female body. You're putting it inside the female body. Fertilization is going to happen inside the body itself. IUI, intrauterine insemination. When you're not putting elsewhere, you're directly putting inside the uterus. The semen you're directly putting inside the uterus, you call it intrauterine insemination. Again, both of them, they are in vivo techniques. They are in vivo. This is in vivo, this is in vivo, and this is also in vivo. In vivo means they are put inside. Okay, they have been done inside. So ZIFT, ICSI and IUT are the only ones, 2, 3 and 4. 2, 3 and 4, your answer for this question is going to be A. Okay? Yes. Which one of the following symbols are used for representing sex chromosomes of birds? Any idea? So female, in the case of birds only, you for the first time you see female heterogamety. Female heterogamety. That means females are going to produce two type of gametes. So over here ZZ is for males, they produce only one type of gametes, all of them having Z type of chromosome. One is ZW, okay, where some of them have produced Z type of chromosome, some will be having W. That is why female heterogamety. Always remember W for women. So here ZW is the one that is there for women, and that is happening in the case of birds. Okay, during the growth of any population, more individuals acquires peripheral character values at both ends of the distribution curve, which leads to what? So listen to me. Growth of any population, more individuals acquires peripheral character values at both ends. That means, suppose this very, what do you call, complicated thing. You have tall, you have dwarf people. When I say you have tall and both dwarf people, what do you say? This is selection of both these values. This is basically selection of both these values. That is what is meant. This type of growth curve is known as disruptional, or basically disruptive growth curve, because both the extreme characters are being selected. If at all I say neither tall, neither dwarf, the middle character is being selected, that is medium heighted, then it is known as stabilizing selection. If I say only the dwarf ones are selected, it means that it is directional selection. So over here you have been asked about both ends. There are values at both the ends, so it is going to be disruptive. C is going to be your answer for this. Next, refer to the given figure showing human fetus within the uterus. How will the removal of A? A is this. A is basically what? C. What is A? First identify it. This is the baby, which is fetus, which is being formed over here. This is going to be the placental villi, that basically means placenta. Over here you have an attachment thing. What do you call this as? This is umbilical cord. Umbilical cord, which is a connection link between the placenta and the fetus. What does it do? It is there for transportation of things, right? So if that thing is not there, removal of A is happened, what is going to be having the effect on the growth of the fetus? That is the question. So first identify it, first know its function, and then look for the answer. Secretion of HCG hormone will stop. Arey Baba, placenta does it. Why will it stop? Placenta is still there. Secretion of relaxin hormone, ovary does it. Transportation of substances to and from the embryo will stop. This is correct. All of this above, no. C is the only answer. Okay, C is going to be the only answer over here. Transportation is going to be hampered. Cool. Tubectomy is a method of sterilization in which, tubectomy is what? The fallopian tube, my dear children. Over here, the fallopian tube that carries the egg is going to be cut. Is basically going to be cut and tied up such that the did I say sperm by any chance? I'm so sorry if I did. Such that the ovum basically is not getting transported. So tubectomy is a method of sterilization in which the fallopian tube is being cut. Small part of the fallopian tube is being removed or tied up. Okay, correct. Ovaries are removed. No. Small part of vas deferens. No. That is vasectomy. Uterus is removed surgically. No. Only answer is A. Got it? Yes. Now, by the way, a question to you guys. You can answer me. Here, menstruation will happen. This is homework question 2. Do you think menstruation is going to happen in this particular condition for those females who have underwent tubectomy? Tell me in the comment section. Okay. Total 512 seeds are collected from the cross of capital W, small w, capital Y, small y, and the same thing. Find the number of plants produced with the first dominant and the second recessive trait. What? So it just looks like capital R, small r, capital Y, small y. When you cross this, what did you get, Baba? You got something like this, 9:3:3:1 ratio, right? 9 of them were showing round and yellow character. Round and yellow. Both of them are dominant thing, and both of them were found in the dominant parent also. Over here, one standard for wrinkled and green. Both of them were the recessive character, and both of them were found in the recessive parent as well. These are the non-parental characters, something that was not there in the parents. Round with green this time. Round is a dominant character, green is a recessive character. Wrinkled was with green, now with yellow. Wrinkled is a recessive character, yellow is a dominant character. Get it? You are being asked, find the number of plants produced with the first dominant and the second recessive trait. First dominant and the second recessive. Here both of them are dominant. Here first is dominant and the second one is recessive. Here first is recessive and the second one is dominant. Both of them are recessive. So you have to basically find about them. So out of 16, 9 plus 3 plus 3 plus 1 is equals to what? 16. So out of 16, 3 of them are like that. But now you have to find it out of 512 seats also. So what do? Basically, if 3 out of 16 gives the non-parallel category that's like this, you have to find it out of 512. You do the calculation, you'll get your answer. It comes out to be, I think, 96. Yes. Okay. Important. Fine. Yes. Now, this is a homework question that I want you guys to do. A hominid fossil discovered, this would be homework question 3 now. A hominid fossil discovered in Java in 1891, having a cranial capacity of about 900 cc was Homo erectus, Neanderthal man, Homo sapiens or Australopithecus. I'll tell you a hint. Okay, before them came Homo habilis with 650 to 800 cc. Now you tell me the answer. After Homo habilis, who is the one that comes? Who has 900 cc? Okay. 57th question. The signals for parturition originates from. The signals that baby has to move out originates from placenta only, placenta as well as the fully developed fetus, oxytocin which is released from the maternal pituitary, fully developed fetus only. What do you think is the answer? Signals for parturition originates from placenta as well as the fully developed fetus. Both of them are equally responsible for that. So I'll say B is your answer for this particular question. Okay, very important. Perfect. Next, intracytoplasmic sperm injection is a specialized procedure in which the sperm is directly injected into the intracytoplasmic sperm injection. Inside the cytoplasm, you are injecting the sperm. Array, whose cytoplasm, Baba? Obviously, it is going to be the ovum, the egg cytoplasm. Then only you will get what? The zygote out there. That is why you do ICSI, no? So your answer for this question is going to be ovum. Cool? Cool. Next one. Which of the following have equal number of chromosomes? Klinefelter syndrome, Down syndrome. Klinefelter syndrome, Turner syndrome. Turner syndrome, Down syndrome. Turner syndrome, Yanomorphy. Listen to me carefully. We just have 3 things in our book. One is Downs, one is Klinefelter's, and the other one is Turner's. Now, in Down syndrome, what is happening? You should have a pair of 21st chromosome, but you have one more extra chromosome, making it. Usually we have 46 chromosome, but one extra chromosome comes together. What do we have now? 47 chromosomes out there. In Klinefelter, you should have had X and Y chromosome, but now you have an additional X chromosome out there. Again, normal chromosome should be 46, but you have one extra X chromosome, making it to be 47 chromosomes. Which is one is same, obviously them two. But let's see. In Turner syndrome, you should have had X chromosome and what? And basically another X chromosome, but here you have nothing. You do not have anything. So it should have 46 chromosome, but now -1, and it will be 45 number of chromosome out there. So which one is correct? Which of the following have equal number of chromosomes, Baba? A. Klinefelter's and Down syndrome will be having equal number of chromosomes. Cool? Yes. And the last question. Arrange the following in the order of their evolution. Homo habilis, Ramapithecus, Homo erectus, Dryopithecus, and everyone out there. First guy to come into the picture is basically Ramapithecus and also Dryopithecus. Dryopithecus basically were more ape-like. Ramapithecus were more man-like. From Ramapithecus came Australopithecus. From them came Homo habilis, the first guys out there. Homo habilis, the first Habibis, yeah? And then from them came Homo erectus. Homo erectus. From them came 2 things. Neanderthal man, okay? And then came Homo sapiens at the end. So, how do you find your answer? First of all, try to find if Homo sapiens are found at the end. Homo sapiens are found at the end. So here I see in this last option, Dryopithecus is the end. So wrong statement definitely. Neanderthal man is there. Okay, so sometimes they might have missed Homo sapiens. So these 2 options are still there. Okay, what about this option? This option is also still there. Okay, let's go ahead. So we have 3 options to look forward for. Now, Homo habilis, can it be the first one? We know that it's not the first one. Wrong. Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, Homo sapiens. This is exactly what we studied. So this can be the answer. Australopithecus, Homo sapiens. Homo sapiens in the middle, Baba. That is definitely going to be wrong. Your only answer for this question is going to be B. Okay, so they were more man-like. They gave Australopithecus. They were the people who made weapons and stone weapons. They did not eat meat. They were vegetarians, pure vegetarians. From them you got Homo habilis. Homo habilis basically were the first organisms to have human-like features, hominid-like features, and they also ate fruits only. And they had a cranial capacity. From them only the cranial capacity started. It was 650 to 800 cc. From them you got Homo erectus, the ones who could stand straight, they erect, and their cranial capacity increased to 900 cc, and basically they started eating meat also. And then from them you got Neanderthal man first, and then you got Homo sapiens as well. Both of them you got from the same people. But yes, Neanderthal man were the first organisms to bury their dead and cover their body, and their cranial capacity was the highest. It was 1400 cc. Okay, this is the basic information which is out there, and with this basically we covered the entire part test of ABHYAS test series for biology. Now tell me in the comment section, first of all, how was it? Do you think it is nice? And also please let me know the answers for the questions that I have asked you, and make sure you take it up at the earliest. I'm telling you, definitely it is going to be helpful. Here I was there to help you out with some questions. Otherwise, I'm pretty sure you might have been stuck at certain questions out there. Yes, so that's all. Wishing you the best, and my dear children, do not forget to hit the subscribe button. We are coming on Monday with something very, very important for every KCET aspirant out there. Yes, wishing you the best. Take care. Lots of love from Jasmine ma'am.